In this brief video, we illustrate how the pcmp app can be used with a collection of nuclei sequenced in the Allen Brain Atlas project. You can work with this app at the shinyapps.io humbr18c. We obtained a stratified random sample of 1,800 nuclei, 300 for each of two regions. Those are the anterior cingulate cortex and the primary visual cortex, and 3 donor IDs. For each nucleus, we obtained the fraction of reads that map to introns for all genes as a cell-specific measure of prevalence of immature transcripts. The quartiles of this measure, which we can find as the last variable here, pim-cort for percentage of immature transcripts, we've taken the quartiles of the distribution of that. They show a remarkable gradient in the PC1 versus PC2 projection. And we can use the simple differential expression technique built into this visualization tool to get a feel for genes that differ in expression pattern between these relatively immature nuclei and those that have fewer reads mapping to introns. And what we see here are genes that are slightly upregulated in the selected group, nuc-7L3, SRRM2, which are annotated to splicing processes. We can go back and change the selection to cells that have fewer reads mapped to introns and get a list of genes that are differentially expressed in that selection. Notice it takes some time for these tables to update because of the large numbers of features involved. We also have access to information about structure remaining in the selections that were made. So, for example, it's possible that in the second selection there's a clustering within the cluster that we selected. So that's just one of the directions we can travel with this tool. Clearly of great interest is whether we can find structure distinguishing cells from the different brain regions and give interpretation to cells identified in that way. And of course we want to be able to compare different projections. So for example, here's a comparison between the UMAP projection, first and second components, and the t-SNE projection, where it seems easier in the UMAP projection to identify a group of cells that comes from the ACC. Not quite so easy with t-SNE. And we can drill further down into deeper parts of this re-expression process by interacting with these displays. Here's one where t-SNE components 4 versus 5, it becomes easier to identify a group of ACC cells, and then perhaps there is some substructure within that collection. So, shinyapps.io, humbr18c, you can interact with these data yourself.